We've got a great panel of speakers, and I'm going to turn it over to our first speaker to kick things off on our session about gene editing. We have Dr. Ray Liu, and Dr. Liu is a molecular geneticist from the University of Guelph. He specializes in gene regulations. He's worked on viruses and cells and lab mice, but fortunately for us, he's also taken a keen interest on pigs and swine genetics in recent years. So I'll invite Dr. Liu up to the stage.
Thanks, Daniel. Also, I'd like to extend my thanks to XIP for organizing this event and also for inviting me to talk about gene editing. That's an area that we move on to with collaborators, with funding agencies, and with my colleague and friends, Vilser Bordignon, who is going to follow up with my talk. So first, I will do a very quick intro, how I got into this swine genetics. So I'm a molecular geneticist by training, did my PhD in Saskatchewan. So my PhD thesis was to generate normal molecular markers to separate some of the leaf-cutting bees. So it's different from my master's work was on human gastric cancer. I did a postdoc at WCVM, work on bovine herpes virus, and that's how I got into viruses. And the underlying theme was stress. What are the genes or stress-related genes that involved in BHV and IVR that affect the animals? Then I went on to NIAID as a research associate, which is a glorified postdoc. The NIAID, so some of you guys may have paid attention, so the director of NIAID is Anthony Fauci, so who is the COVID guy on TV for the past few years. So I did 2 years of that and recruited back to Canada, and I became a faculty. And during this time, I identified 2 genes. So one is we call lumen, the other one is called zhangfei. 2 Chinese warriors that want to play video games and came across. So these are the 2 genes we named. And later on in Guelph, my first PhD students identify a regulator for the gene regulator, so which we call ARRF. Interestingly, so these genes are involved in stress regulation and also regulate metabolism, which is not surprising. So when we made gene knockout for these 2 genes, the mice are lean and no stress. So these are the 2 phenotypes that are really striking, and that's got me motivated to see how we can apply this knowledge to human medicine or to agriculture. So that's how I got into a swine business. So in recent years, we are doing gene editing in cells, and also we are collaborating with Vilser, try to move on to swine genetics. So in case there are audience here that not familiar with genetics background, this is a very fundamental stuff. So DNA makes up all these, letters, very long. So we have something around 20,000 genes. For every gene, we have 2 copies, which will come to play later on. So human genome and the pig genomes, similarly, about 3 billion base pairs. 3 billion base pairs, or these letters, ATCG. So there's just billions of them. And of course, variations exist. So not only they exist between individuals, you and me, between pigs, but also exist between cells in the same organism. So variance exists naturally. That's one message I try to convey. Secondly is that the vast majority of these variations play no role in terms of a phenotype or impact on the cell or the organism. So if you pick up any base substitution or any variations between, let's say, A and T, the outcome can be nothing, like the AGA changed to AGG, it still codes for the same amino acids, so it does not change. Or can change a little bit, change from one amino acid to a different one, from arginine to serine or arginine to lysine. So these two amino acids are very similar in terms of the function, and these two amino acids are different. Or you can pretty much kill the protein because once you mutate AGA to TGA, you create a stop in this protein synthesis. Basically, the gene very likely will be dead. So therefore, the gene variations or DNA variations exist naturally. A single letter change can have drastic effect or no effect at all. If this change or variation happens in the germ cells, or the cells will give rise to sperms and eggs, you will be able to pass along to the next generation. So that is the basis for all the genetic breeding, right? So one example I dug up, and the audience probably know way more than I do, and they found that the weaned pigs per cell has doubled in the last 50 years because of the breeding, right? So it's very effective. So moving on forward, though, traditional breeding may face some hurdles. Big improvement may get harder because after decades of selection, some variations are rarer than others because it just doesn't occur because just pure genetics. 1 in 100, 1 in hundreds of thousands chances for certain variation to occur. Certain traits have not been selected for, such as novel pathogen resistance, right? Never exposed to the pathogen, so therefore never has been selected for. And of course, if you found a trait in a pig or in another herd, it's difficult by traditional method to introduce to your own breeding herd. So that basically set up the basis for gene editing. So what is gene editing? So originally discovered in bacteria. So the CRISPR is short for a particular DNA sequence identified in bacteria. Cas is the enzyme that characterizes this work. So often people just call CRISPR gene editing. The full name should be CRISPR-Cas. So what it is that this protein will generate a cut on the DNA, and then when the cell tries to repair it, you often create deletions or insertion in the DNA and cause the gene will be disrupted. So that is the very origin of the gene editing tool. Now the technology has advanced a lot. So again, Vilsa will talk more about that topic. But today, though, gene editing can be a precise genetic toolbox. So you can introduce at a single letter position to change a particular letter to the other. So therefore, you can do a one-step breeding to introduce a variation or trait to your own breeding herd. So that's the power of this technology. The other thing is that at the DNA level, the difference or variation introduced by the gene editing will be exactly the same as the variation happening in nature. So this is something that probably many of you guys have heard. These are PRRS-resistant pigs. So genus PIC, they have these pigs, and I read about it. So they actually are betting on the pigs will be able to be marketed by 2025. Some of their spokespersons say that on the news. I don't know when that day comes into play, but they are confident that we'll be able to marketing that, and they actually have bred something like thousands of PRRS-resistant pigs in their barn already. So the way they made this is by disrupting this particular gene or protein called CD163, which is a surface protein that allow the virus to get in. So you see this one. If you eliminate CD163, the virus cannot get into the cell. So therefore, the pig will be resistant to this virus. So you introduce a mutation and knock out CD163, and your pigs will be immune to this virus. So that's the background of gene editing and viral resistance for the PRRS. But for African swine fever, though, the question is more complex. So first of all, some background. So they affect domestic pigs and wild pigs equally. There's no difference in terms of which ones affect better or infect better. Highly contagious, high mortality to naive or unexposed pigs. No treatment available yet. And these viruses are very stable on the environment, so in a wide range of pH, persist in environment for days, in meat product for months, in vector insects for years. So that's reason why it's so hard to get rid of this virus. Of course, it will pose a tremendous risk to farmers if they detect it, right? So once it happens in a wild boar or close to the barn, and the export market will be shut down. So that's what happened to Germany a couple of years ago. So in terms of ASFV transmission, so the wild boar part people know pretty well. Through dead animals, contaminated environment, feed, and of course this insect vector, so these soft ticks. So they bite infected pigs and bite other pigs, and the virus can sustain in these ticks for years. So even there's no infected pigs around, and the virus can survive in the insects for years and then bite another animal, come back again. For domesticated animals, their internal transmission is pretty much similar, right? So you have pig-to-pig transmission through feed and from human contamination. So the mystery is how it gets this virus from the wild boar, which is still unknown. But there are many evidences that suggest that human activity is a main part. Feed as well. So contaminated feed, like even they're talking about maybe crops, if they were fed to the animals, can bring. But human activity is probably one of the key contributing factors. So that brings the questions, the wild boar. So this is a big problem, is the most prolific invasive mammal in Canada. So this is a map that generated by Ryan Brook in the University of Saskatchewan. So this is the survey data until 2000, and this is 10 years later. So these invasive species have been highly successful and expanding territory really, really fast. So just a matter of time that you will get to an area near you. So it caused a lot of damage, estimated by USDA about $2.5 billion to the US. Environmental ecosystem, and most importantly to us, is they are reservoirs for pathogens, bacterial diseases, swine influenza, so that's dangerous for human, and of course it's ASFV. So how we tackle the problem? There are many ways people have tried and failed miserably, hunting, trapping, even poisoning, and some areas even experimented with contraceptives. They release these pills to the wild and make the pigs sterile, whatever. None of them worked. So in Texas, people spend hundreds of millions of dollars and try to shoot those wild animals. And I think the best thing they have achieved was 23% reduction in the wild boar population. But you need something like a 75% reduction to really have an impact on the population growth. So that brings the topic of gene editing. So one thing that people have tried right now and still expanding is using gene editing-based technology called gene drive in eradicate malaria. So they basically have tried to gene edit mosquitoes. So these are the vectors for this pathogen. Two strategies: one, modify mosquito to make the female sterile. So in a way to collapse the mosquito population. So in an isolated area, people actually can successfully kill off all mosquitoes. Of course, this is a very ideal closed environment. So this is talking about gene-edited allele frequency, like how many edited allele exists in the mosquitoes. So you can see drop to 0. But that's actually highly unlikely in the wild. The other strategy is to make the mosquito that unable to transmit the malaria pathogen. So you can see in the end, if the drive allele or the gene variant is penetrating the entire mosquito population, so you will be occupy probably half of these insects. So that will be that. So the two strategies have been used in tackle malaria, and people are expanding this. I think as of this year, those mosquitoes were released in California and Texas. So it has been approved by FDA or by the US. So that basically gives us the idea, can we use a similar strategy to control wild boars? So first I want to introduce a so-called gene drive concept. It's not entirely new. So basically, people want to create this gene variant, what we call alleles, that actually can self-propagating. So normally, if you have one gene variant allele, so your mate and this one allele will eventually dilute it out, right? Some variant popped up. But as the animal proliferate, and this frequency will dilute it out. But if you engineer a gene variant and make that gene variant has the capability to convert the other copy, because the animals in us, we have 2 copies of every gene. So when you engineer one copy and the engineered copy can convert the other copy to itself, so now you see the impact. So edited, non-edited, in next generation, so you will convert the next generation to itself. You see that? So you actually can self-propagate its allele. So this is so-called a gene drive. So this is something that I noticed that released earlier, actually last year now, so in the summer by USDA. So what they were proposing to do is exactly what we had in mind, is use gene drive to control rodent or pest species, where using gene drive or gene editing to suppress invasive rodent, basically the offspring will end up with only one sex. So each generation end up with one sex, that eventually this population will shrink or hopefully die off. So either sterility or a single sex offspring. So come back to this chart. So the strategy is to create a female-specific embryonic lethal. So all the progenies that produce are all male boars. So male boars can go out and copulate and spread this allele. and females basically are die in the womb. So that's one way. The other way is the female sterility. So all the female borns are sterile, and males born can still produce a sperm, and sperm carry this allele, continue to spread in the population. So that is the thought that's encouraged and helped by SIP. So we have a study, a feasibility study on the way. It's an on-paper exercise. We look at the genes, look at the molecular designs to see whether this strategy will be feasible combined with computer simulations for that. In terms of a safety considerations, so something that we have discussed and talked to colleagues. So first of all, I want to emphasize that engineered alleles can only spread by mating. So this is not a virus where the DNA and the RNA can self-replicating. So it can only spread by mating. It's almost like, can you spread your red hair to your neighbor? You cannot. So in your gene, in your genome, it just cannot get out. No higher risk for wild boar farmers, in case that's a concern for some of the people who have wild boar farms. This gene drive will work, will perpetuate the wild boar population or domestic pigs equally. Will not wipe out wild boar population instantly, will take generations. Off switch can be built in this gene drive system, so we can turn it off if required. And also we thought about to engineer a tracing system where we can trace or identify the pigs that somehow got this allele in the genome. So basically that is the design that we are working on at this time. And with that, I will just thank my funders over the years. Of course, Swine Innovation Porc for helping with this idea, sponsor for the project. And my venture into swine genetics, Alliance Genetic Canada, which we worked very closely in the past few years, and I really appreciate for their help and support. And CCSI, Canadian Center for Swine Improvement, that helped me got into this swine business, and I really enjoyed it. Ontario Porc, provincial government, AAC, and NSERC. So with that, I'd like to answer any questions you may have. Thank you.